Now we'll talk about the steps of the division of the nucleus. We'll talk about the steps of mitosis. Cell division is made up of two steps: mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. So these two steps together make up cell division. So it's important to emphasize that cytokinesis is not one of the phases of mitosis. Now, mitosis, it's described as either having 4 phases or 5 phases, depending on which textbook you're looking at. But those phases are prophase, metaphase, anaphase, and telophase. In textbooks that only have 4 phases for mitosis, prophase is often divided into early prophase and late prophase. For those textbooks that use 5 phases, it's that late prophase which is known as a step called prometaphase, sort of between prophase and metaphase. So let's talk about the events that are occurring during each of these phases, starting with early prophase. So a cell that's in early prophase, it has completed interphase. It's copied its DNA. It has all the machinery it needs to divide. In the first steps of prophase, what begins to happen is that the chromosomes start to condense. The centrosomes, the structures that organize the microtubules, they were copied during interphase, and now they start moving apart. They move towards opposite poles of the cell. We use the term poles to describe 2 different ends of a spherical object, much like the Earth has a north pole and a south pole. So by moving to opposite poles of the cell, it simply means opposite sides of this spherical object. Mitotic spindle begins to form. The mitotic spindle is made up of microtubules coming from the centrosomes, and we'll see this mitotic spindle will play a role in separating the chromosomes during cell division. So these are the events of early prophase. In late prophase, the nuclear envelope breaks down. The mitotic spindles extend from the centrosomes and attaches to the centromeres of the chromosomes. If you recall, the centromeres are those connection points between the 2 sister chromatids of a duplicated chromosome. And in fact, there's a specific region on those centromeres which the microtubules attach to, and those are the kinetochores. This is going to happen with microtubules coming from each centrosome. And so in this way, each of these duplicated chromosomes are going to be connected to both poles, and the sister chromatids are going to be linked to opposite poles. One side of each duplicated chromosome connected towards each pole of the cell. Once all the chromosomes have been attached to microtubules, the microtubules will end up organizing and pulling all those chromosomes to the middle of the cell. This step is known as metaphase. And there's a line down the middle of the cell where these chromosomes are lining up, and that is known as the metaphase plate. During metaphase, all the sister chromatids line up along this metaphase plate. All the chromosomes are present, and the sister chromatids are equal and balanced, ready for segregation. Segregation occurs during the next phase of mitosis, known as anaphase. In anaphase, the sister chromatids separate. The sister chromatids break free and are dragged to opposite ends of the cells. The reason why this happens is because the centromere that's holding those two sister chromatids together gets split down the middle, and this is due to the microtubules shortening. The result is that each of the duplicated chromosomes has now been split down the middle, with one sister chromatid traveling towards each pole. Once the sister chromatids separate from each other, they are now known as daughter chromosomes. We have the daughter chromosomes moving towards opposite poles of the cell. The final stage of mitosis is known as telophase. During telophase, chromosomes reach the opposite poles. The mitotic spindle begins to fall apart. The chromosomes begin to decondense, and the nuclear envelopes reform around each group of chromosomes. Typically, around the same time as telophase, cytokinesis begins. Remember, cytokinesis is not one of the phases of mitosis, but cytokinesis is the division of the cytoplasm. So with completion of mitosis with telophase and completion of cytokinesis with the division of the cytoplasm, we have now successfully completed cell division. Two cells have formed, each with their own nucleus. Now, as a quick recap of the events of mitosis, a student shared with me a memory aid that he used to remember the general steps and order of mitosis. And it has to do with the first letter of each phase. P, prophase, well, that stands for prepare. This is the cell preparing for the division of its chromosomes. So the chromosomes condense, the nuclear envelope breaks down, the mitotic spindle forms. Those are the steps in prophase. Next, metaphase starts with M, and that stands for middle. That's when all the duplicated chromosomes are lining up in the middle of the cell, on that line known as the metaphase plate. Anaphase, A, well, that stands for apart, because that's when the sister chromatids separate from each other, becoming daughter chromosomes. They move apart towards opposite ends of the cell. And then lastly, telophase, well, T stands for ta-da, because it's the end, it's the last step. The nuclear envelopes reform, the chromosomes decondense, and we have a divided nucleus. Again, this happens around the same time as cytokinesis, and so the end result is we have two daughter cells, each with their own nucleus and with their own cytoplasm. So there we have it, the steps of mitosis: prophase, metaphase, anaphase, and telophase. In the next video, we're going to talk about what happens when cell division is not controlled, and this is a specific disease known as cancer. See you in the next video.